Welcome to BioClass. Today we will discuss translation, the last phase of protein synthesis. Translation, already we have got genetic code, and from that we know that which triplet code will produce what type of amino acid. Here if again we can see that it means the translation now according to the sequence of mRNA that how that amino acid will form. Suppose UG, if it is there in that mRNA strand, then leucine will produce, or CCG, if it is there, then protein will produce. CAG is glutamine will produce. So now it is clear. So if I give you one sequence and this dictionary of that is genetic code, then easily you can find it out that according to that the triplet code, any sequence if you make, then also you can get one protein, amino acid chain polypeptide, which is protein. Now translation, here it is a process. Now this process we have to discuss how this process, how it can perform and where it can perform. So all these things it will be discussed now. Now look at here, the first in this we have to keep the concentration. The amino acids are joined the bond with known as the peptide bond. And that peptide bond that already have, the formation of the peptide bond requires energy. Therefore, in first phase itself, the amino acids are activated, the presence of ATP and linked with tRNA. The charging of tRNA, aminoacylation of the tRNA, that we have to discuss. Okay. Now, tRNA is transfer RNA. tRNA will carry anticodon, and as well as it will carry that amino acid. So when it can carry, when it will be charged, when the charging will take place with this aminoacylation process will take place, then it can carry. It means that 20 different type of the tRNA will be there, the raw material to produce the protein. So we can see this. These are so tRNA structure that you have to in this tRNA, here will be the amino acid. What amino acid means? Here the amino acid which is here. So that any of the amino acid, here it will be. Here is the adenosine triphosphate. This process requires energy. Here the ATP. ATP here when it will join, it will form that aminoacyl EMP, adenosine monophosphates. The 2-pyrophosphate. This is ejected. Then after that it joins in free end of the tRNA. Okay, free end of the tRNA. So this joining we can see any other way also. Look at here, the valine, here aminoacyl synthetase, this is an enzyme, and ATP here joins, and uncharged tRNA, here it is comes and it turns into charged tRNA. Look at here, charged tRNA will going to add this valine in the structure. So valine here come here in the concept of that codon anticodon GUU. So this is codon. You can see this, the GUU will produce the valine. So GUU, this coding, it will be in mRNA, and whereas this tRNA will carry this anticodon. So codon and anticodon is complementary. So if it is not, then this tRNA will not set there in the mRNA. Okay. So GUU, according to that, it is valine will produce. So I mean valine already it is added here in this. So here it is. When it will come here in the mRNA with the anticodon. So GUU, what is the anticodon of GUU? CAA. You can see that it's complementary part of it. All right. Now come here in process of translation. Here also they are divided into initiation, elongation and termination. There are three clear phases. It is, and the component we are going to use that again here I am showing you. This is mRNA. This is mRNA, the processed mRNA that already in transcription we saw that this is the cap, this is the untranslated region UTRs, this is the start codon, this is the stop codon, this is the poly-A-tail that already you have I think studied that part. Alright, now this is mRNA. mRNA you have to remember that mRNA it is produced in nucleus. From the nucleus, this mRNA it moved out through the nuclear pore, and it, now in here it is in cytoplasm. Here it is a ribosome. I think in prokaryotic, eukaryotic, you know that ribosomes are different. So here I am not showing, I am showing in general. Here this is the small subunit, and this one the largest subunit. The smallest subunit here in case of eukaryotic it is 40S and larger it is 60S in case of eukaryotes. Okay, both are very important. Now look at in this initiation factors. Initiation factor, it means when the process will initiates, the initiation factor. Sometime in case of eukaryotic we are writing EIF1 like that. So E stand for eukaryotic initiation factor 1, 2, 3 and more are there. So for your standard that is this 3. And elongation factor here, and here it is terminator. I mean termination, not actually terminator, it is I mean it is found in at last, that I will tell you. Now these are charged mRNA, tRNA. This is the tRNA, charged tRNA. Charged tRNA here this is anticodon. According to that they will search that codon. If they will get that, they will add the amino acids there, otherwise not. So this is anticodon and this is amino acid. So in that way they will search. For the searching this factors will help. The ribosome will help. In ribosome, the large subunit, you can see that the peptide sites and amino acid sites. This is the peptide, this is the amino acid site. So in brief these are there. Now I'll show in this and in video so that you can understand. Look at here. This one the processed mRNA which transcribed there in the nucleus, and after that it came here in the cytoplasm. Here the initiation factor 1 and 3 it separates this larger and smaller subunit. Now smaller subunit, it adds to the cap region, not UTR, actually it is cap region, and here that UAC will search the first codon, I mean EUG, initiation factor 2 also added, and after that it will start the process. So this whole area it will search, and when it will get that it will fix. When it will fix, then the large subunit will join, and that UAC contains methionine, which is the start codon. I mean UAC is anticodon, AUG is codon. Now this all initiation factor, it is removed. So initiation process is completed. Now next, here the amino acid site, here one by one it will come. For that elongation, next part is elongation factor. Here it helps to enter inside the amino acid site. So GUU will carry that anticodon. Now you can see that the anticodon, it comes with the tRNA, carries the anticodon, and the valine here, the amino acid valine. So you can see the CAA, GUU, and the valine and methionine. Now methionine breaks, and the tRNA will eject, and it will join here the peptide bond, methionine with valine. Methionine and the valine will join the peptide bond. The next they will shift here in amino acid site to peptide site. You can see that this chain is now 2. Next amino acid GGG, here the codon. For the GGG, here it comes CCC anticodon containing tRNA and with glycine. Here again that peptide link will break from this old tRNA, and here the chain will form. Now this chain, it is 3, the methionine, valine and glycine. Next it will produce UCA is the codon, and for that the anticodon, the tRNA will bring with serine. And this is the tRNA with the anticodon and the serine. Again the glycine, here the peptide bond will break, and it will join to the serine, and they will form the peptide one. So peptide site CCC, that tRNA, it will eject. Next it will shift it to the peptide sites. Now you can see that the number of the amino acid is 4. Now UAG will form. UAG, we know that it is stop codon, so that the no tRNA will come. One releasing factor, RNA releasing factor, it is there, which will release this from the tRNA to the amino acids chain. Now it is polypeptide, so everything is dissociates. Here ribosome again gets their own shape, and here the newly formed that amino acid. Now you know that the ribosome, the endoplasmic reticulum, it can be stored. So this is the process. If we see the points here, all the points that just now we discussed, that initiation here, that translation takes place in a ribosome inside the cytoplasm. So that you have to keep in mind. Before that, the transcription, it was in nucleus. mRNA, it moved out the nucleus and reached to the ribosome, to the nuclear pore. Initiation factors, there are several initiation factors, you can search there. Joins the small subunit and it separates from the large subunit. Now mRNA with here I mean amino acid, it joins the small subunits, amino acids, and IF2 it helps the process. tRNA with anticodon and methionine, it joins this first one and the methylated cap, that region, and it starts searching that AUG and fixes AUG, and methionine already it is there. Large subunit now joins to the small subunit, that is the last phase of the initiation. Elongation, you know that it can be very long, but in your discussion it will be the shortest one. The tRNA now binds to the amino acid site to the ribosome. Here elongation factors, it helps to join. The codon wise now it is added, which will be carried by the tRNA. Methionine is first, after that the one after another. Now the peptide bond will make. Empty tRNA ejected to the site. And there are some book, there are 3 sites, but according to your book that I kept it in 2 sites. Termination, when the mRNA stop codon it appears, that is UAG, UGA, UAA, then process will terminate because no tRNA is there, no tRNA will carry anticodon or amino acids. RRF, it helps to cut that last amino acid to tRNA, and so it is separated, and amino acid now it is stored in endoplasmic reticulum. Okay, so this video also you have to see several times to understand this fact and write it accordingly. Thank you.